Welcome to The Explainer. Today, we're diving into what I can only describe as a biological true crime investigation. We're going to be tracking an incredibly stealthy, microscopic killer, one that has totally mastered the art of infiltration and evasion inside the human body. So settle in, because we're going to break down the highly calculated and honestly deadly biology of Amoebiasis.
100,000. That's a staggering number, right? Well, that is the exact number of global deaths caused every single year by the parasite we're examining today. It doesn't have claws, it doesn't have teeth, yet it operates with this devastating efficiency, quietly taking lives on a massive scale year after year.
And that brings us to the central mystery of our explainer. How exactly does this parasite, Entamoeba histolytica, which is actually the second leading cause of mortality due to parasitic disease in humans, operate with such lethal precision? How does a single-celled organism completely outmaneuver the complex human immune system? Well, to answer that, we first need to look at the battlefield.
Section 1. A deadly global threat.
The sheer reach of Entamoeba histolytica makes it a massive global health crisis. We're talking about roughly 50 million cases of colitis or extraintestinal disease worldwide. It thrives particularly in tropical countries with warm climates. In these areas, things like contaminated water, poor waste management, and faulty drainage systems basically create the perfect storm for transmission.
What's truly wild is how drastically environmental factors swing these infection rates. When you break down the prevalence of amoebiasis among school-aged children across different geopolitical zones, the disparities are just huge. In Zone 1, you see a 16% infection rate, but jump over to Zone 2, and that number skyrockets to nearly 68%. It perfectly illustrates how this parasite ruthlessly exploits vulnerabilities in sanitation infrastructure.
Section 2. Anatomy of the invader. Let's shift our focus down to the microscopic level to examine the biological weapons of our antagonist. Entamoeba histolytica is essentially a shapeshifter. It exists in two distinct morphological forms. First, you have the cyst. Think of this as the armored transport vehicle. It's small, it's stable, non-motile, and it has this incredibly tough refractile wall containing chitin. That wall allows it to survive the harsh acidic environment of the human stomach. On the flip side, we have the Trophozoite. This is the active attacker. It's much larger, highly motile, and pleomorphic, meaning it can actually change its shape. But it has one massive weakness. It is completely unable to survive outside the human gut.
So meet the active Trophozoite form. This is the replicating phase. You've got the cytoplasm and that distinct circular nucleus with a karyosome right next to the endosome. It reproduces by binary fission and gets its energy by literally gobbling up bacteria and food particles. Make no mistake, this is the form that actively invades and causes severe tissue damage.
Contrast that with the stable cystic form. It's got a tough exterior, a distinctive chromatoid body, and glycogen stores packed inside the nucleus. This right here is the parasite's survival pod. Because the active trophozoites would die almost instantly in the outside environment, the parasite insists itself to survive in stool, waiting patiently to contaminate water or food and ultimately find a brand new host.
Section 3. The cycle of infection. So how does this survival pod actually get inside us? Well, the invasion is a highly organized four-step plan. Step 1 is direct contact, typically fecal-oral through the ingestion of cyst-contaminated food or water. In Step 2, that tough cyst survives the stomach acid and makes its way down to the terminal ileum or colon. Here, it undergoes excistation, basically breaking out of its shell to become that dangerous Trophozoite. Step 3 is the attack phase. The Trophozoites invade the gut mucosa to absorb nourishment. Finally, in Step 4, they form cysts all over again in the colonic wall, triggered by a specific protein binding process, and are passed in the stool to start the whole nightmare over again.
And this cycle, it just constantly loops. A healthy person swallows the cyst, it turns into the tissue-destroying Trophozoite, causing amoebic disease, and then it packs itself right back into a cyst to be passed back into the environment. The main takeaway here is how this loop turns totally asymptomatic individuals into hidden carriers, silently spreading the disease through their communities without even knowing it.
Section 4. Symptoms and tissue attack.
Now we're getting into the really destructive phase of the disease. What actually happens when the invader starts dismantling the host from the inside out. The arsenal of the Trophozoite is genuinely grisly. To invade, it secretes proteolytic enzymes, specifically a type called histolysin, which relies on cysteine proteases to literally digest the extracellular matrix of your colon and liver.
It destroys your cells through contact-dependent necrosis. When it breaches the large intestinal wall, it creates these very specific, classic flask-shaped ulcers. And if left unchecked, it doesn't just stay in the gut. It can metastasize to the liver, the lungs, and even the brain.
All of this microscopic violence leads to severe, real-world macroscopic damage. The gross pathology is striking. You get these actual physical amoebic ulcers forming right in the intestinal mucosa because of that necrosis we just talked about. And under the microscope, you can actually catch the culprit in action, pulling off a process called cytophagocytosis. That's a fancy way of saying it's literally eating the host's red blood cells.
But here's the scary part. The threat doesn't stay local. The trophozoite can break right through that protective mucus layer, digest the epithelial cells, and penetrate deep into the submucosa. And once it's there, it hits the jackpot, the circulatory system. By hitching a ride on the portal vein, these microscopic invaders can travel far, far beyond the intestine.
Yep, the gut is sometimes just the beginning. The brutal reality of an extraintestinal infection is the amoebic liver abscess. Hepatic amoebiasis is the most frequent and, frankly, the most dangerous complication when the infection moves beyond the gut. The trophozoites travel from the blood directly into the liver, creating these massive, incredibly painful abscesses. We're talking severe fever, intense right hypochondrium pain, and they can even rupture into the pleural or peritoneal cavities. With a parasite this destructive, you have to ask, how do we stop it?
Section 5. Diagnosing the microscopic killer.
The laboratory detective work required to diagnose amoebiasis is definitely multi-layered. First up, there's microscopy, where we use fresh stool wet mounts or permanently stained preparations, like trichrome stains, to spot the cysts or trophozoites. Then you have serology, using indirect hemagglutination or EIA tests to detect specific antibodies or antigens. And finally, advanced molecular methods. But you might be wondering, why do we need all of these different techniques just to find one bug?
Well, because of an absolute diagnostic nightmare. Entamoeba histolytica is morphologically identical to a completely non-pathogenic, harmless intestinal cousin called Entamoeba dispar. Under a standard microscope, they look exactly the same. You literally cannot tell the killer from the innocent bystander just by looking at their shape or size. So scientists are forced to rely on immunologic analysis and advanced molecular testing just to tell them apart.
But there is one ultimate smoking gun in this microscopic crime scene. If you are looking through a microscope and you see a trophozoite with dark inclusions inside it, which are ingested erythrocytes or red blood cells, you've caught the killer red-handed. That process, erythrophagocytosis, is the only morphologic characteristic that can definitively differentiate the deadly E. histolytica from the totally harmless E. dispar.
As we wrap up this explainer, we're left with a critical question. We've mapped out its biology, we've seen its brutal attacks, and we know its sneaky disguises. But as Entamoeba histolytica continues to exploit gaps in global sanitation, claiming 100,000 lives every single year, will advancing molecular diagnostics finally allow us to outsmart this ancient shape-shifting killer before it strikes? It's an ongoing battle between human science and microscopic invaders, and honestly, it's a battle we absolutely must continue to fight. Thanks for learning with me today.